The material in this communication is subjected to Malaysia Copyright Act 1987. No any part of this communication may be modified, copied, distributed, retransmitted, broadcasted, displayed, reproduced, published, licensed, transferred, sold or otherwise distributed in any way without prior written permission from the owner.
Before we really go into the content itself, let's get started with a quick survey. I would appreciate if you can go to this www.menti.com and enter with the code. So this is just for me to understand where you are, okay? Whether you have any experience before with protein purification, or even if you have no experience at all, it's fine, okay? So at least I know where you are, then I can just go with my pace, okay? Okay, good. We have somebody with protein purification before. Well, as we go through, if you have any questions, feel free to type in the chat box. Okay, so from the response so far, it seems like quite a number of you would not have any protein purification experience before. It's fine, okay? It's totally fine. So working on protein purification is not something that's very tough, but it really needs some passion in it. Okay, so we will go into the protein purification workflow, okay, which is what you need to do after you have expressed your recombinant protein. So what we are focusing today is on recombinant protein purification, but of course the similar principles will apply if you would like to purify any proteins, okay, from any samples. So let's get started, okay? I'll just turn on my laser pointer so that you can follow. The whole objective of protein purification are to isolate a single target protein from a complex mixture, followed by the characterization of the function, the structure, as well as the interaction of the target of protein of interest. I will call it as a strategy in protein purification workflow because we need to go through a series of plans to achieve the overall objective. So such strategic planning includes first the sample preparation. Secondly, we need to optimize the protein isolation condition. Third, we need to strategize the purification method. So in our context, I would recommend to use chromatography techniques in purification, right? So we will also look at whether you want to choose a manual chromatography techniques or you would prefer an automated chromatography techniques. So this depends on whether you have the facilities to do so. So whichever way is not an issue. Last but not least, that is very important, we need to monitor the protein purity as well as to determine whether your target protein of interest still retain its function as well as the activity. So we can observe the purification strategy as first the sample prep. Okay, we extract the total proteins from the cells and we prepare a clarified sample. So when we move on to a higher level of protein purification process, we aim to capture the target protein. So what we will do here is first we have to isolate the target protein. We need to concentrate the target protein as well as to stabilize the target protein. What it means by stabilize here is to ensure your target protein still retain its native structure as well as its activity and function. So during this capturing step, of course, your target protein will not be pure, but it's fine. What we want is to get the protein out from the impurities. So as we move on to next level of protein purification, what we will have here is an intermediate purification whereby we would like to remove bulk impurities such as any other undesired proteins or any untargeted impurities. Lastly, we would like to have a fully polished sample, okay, to remove any trace impurities to achieve the higher level of purity in the target protein of interest. So of course, the more steps we go into, at the end, we will get a much higher purity of the sample, the proteins that we want. But do bear in mind that the more steps that we go into, okay, the more steps that we progress, there will be some protein loss. This is normal and it's fine if your protein loss is not much, okay? It is acceptable. So before we actually start, just an introduction to the proteins, okay? So as far as you may know, there are 4 levels of protein organization. So primary structure of the protein is actually the simplest level of protein organization. It represents the sequence of amino acids that starts with N terminal and ends with the C terminal. So this amino acid sequence is important in protein identification, which can be attained by mass spectrometry or N-terminal sequencing. Okay, moving on to a higher level of protein organization, we have secondary protein structure. So the most common will be alpha helix, beta pleated sheet. So this secondary structure is actually the local folded structure formed within a polypeptide chain due to the interaction between the backbone of the polypeptide. Okay, so it's important that we have to know the secondary structure. Why? Because it tells us about the correct protein folding. Correct protein folding is very important to ensure your target protein still retains its function and activity. So when we move forward to a much higher level of protein organization, we will then have tertiary protein structure, which is the overall 3D structure of a polypeptide that is formed upon interaction between the side chain of the amino acids. So we have 20 amino acids discovered so far. They are all different by their side chain. Okay, so when this side chain interact, they will form a globular shape, which is the tertiary protein structure. And most of the time, the tertiary protein structure will be the functional structure of most of the protein. So as long as the protein retains its native states in tertiary protein structure, it should be functional. Okay, and within this tertiary protein structure, the structure of this protein is maintained by mostly the non-covalent interaction, which is the hydrophobic forces. Okay, so it's important that we know our tertiary structure, our target protein of interest still retain its native states to ensure the functions and the activities still retain. So of course, some proteins can acquire a quaternary protein structure. This happens when those proteins consist more than one polypeptide chain. They can exist as more than one polypeptide chain, one subunit. They can exist as dimer, trimer, or tetramer. Okay, so this is important for you to know what is your protein, what is the target proteins that you are working to work on. So each protein has its own unique shape and maintained by different non-covalent forces. So if the pH and the temperature of the protein environment is changed, or if the protein is exposed to different chemicals, then it can disrupt the interactions that hold the proteins in its native state. So we can say that the proteins are sensitive to pH, to temperature, to the presence of any divalent, monovalent ions. It is important to pay close attention when working with proteins. In most cases, proteins are more stable at low temperatures than a room temperature. Therefore, most purification steps are performed at lower temperature, okay, recommended below 4 degrees Celsius, and it has to be stored at the temperature as low as possible. We will start with our next activity in a short while. So before that, I would just like to tell you that different proteins have different purification strategies, but what do we need to know? So by observing these 4 different types of proteins here, okay, so from visualization, we can tell that, okay, they are different in shapes, okay, but what else we need to know? So the question here is, do you know the properties of your target protein? Okay, let's say you produce a recombinant proteins, but do you know the properties of the proteins that you are going to work with? Okay, when we talk about the properties of the protein, there are two very important parameters that we need to consider. Okay, okay, first, the mass of the protein, which is the molecular weight of your protein. So the molecular weight of a protein can be expressed as Dalton or kilodalton. Secondly, you need to know the isoelectric point or the PI values of your target protein. So the PI values of the target protein will tell you whether your protein is positive charge, negative charge, or they are neutral at different pH range. Third, you need to know whether your protein exists as oligomer or do they have more than one subunit. So this is important so that you want to know whether your proteins will retain the native structure or not. Because during the purification process, you do not want your target proteins to denature. And if there are any presence of prosthetic groups or ligands, so such prosthetic groups can be probably like metal ions, which can be very important in enzymes because some enzymes will require metal ions as the core factor to function. Or if there's any ligands, okay, for example, in the case of glycoproteins, okay, the presence of carbohydrates. So all this will then determine your purification strategy. So if let's say there are presence of ligands, you can make use of these properties to help you in the purification process. Okay, so the primary structure of the protein will tell us about the mass, okay, the molecular weight, as well as the isoelectric points of the target protein. So this is what we are going to test out, okay, using a tool to find out your molecular weight, the PI of your target protein of interest. So let me just share the link with you. Okay, I will just share this link to the chat box. Okay, so you can actually open the link and download the document as Word file. So we are going to use this worksheet to check, to find out the molecular weight and the PI value of the target protein. Okay, are you able to access the link? Okay, so it will lead you to this worksheet. All right. Okay, I'm sure everyone can access to this link. Okay, you can download your file and work on this. So this will be a guideline for you. Okay, what I'm showing here, okay, if you can see my screen, I'm sharing you the screen of the document. If you can see the screen, please just let me know, yes or no, okay, before I proceed. Okay, so what I'm going to do now is, I'll be just using an example, okay, just an example. You can always try with your own protein, no worries, okay? I'm just show you how you can use this tool to determine, okay, just the theoretical molecular weight and the PI value of the target protein, okay? So the example that I show here, oh, sorry, thanks for letting me know. Okay, I'm just going to, okay. Okay, now can you see my document here? All right, thanks. Okay, so what I'm going to show you is an example of alcohol dehydrogenase. Okay, you can download this file and work on your own. Okay, I'm going to introduce you this tool, okay, known as Compute PI MW tool. Okay, so I'm just going to open this link. Okay, so it will brings you to this website. Okay, so this is the website developed by Expassi. Expassi has a lot of resources, tools for proteomics or proteins research. Okay, if you have free time, you can explore this. But what I'm going to show you today is to determine the PI and the molecular weight. So what you can do is, let's say you know the molecular weight, you want to know the molecular weight and the PI of the target protein of interest. What you need to do, okay? Of course, you know the primary structure of the protein. This is the basic thing that you have to find out. Okay, so your target protein, let's say I'm going to show you alcohol dehydrogenase. What you can do is very simple. You just need to copy this sequence and then you paste it here in this box. Okay, so next, I'm going to show you that I will be using average resolution. So why am I using average resolution? So this average and monoisotopic is actually the mass, okay? The resolution of the mass in mass spectrometry. So the reason why I choose average is because in average mass, the mass is average from all the isotopic atoms. Okay, if you choose monoisotopic, it will only measure the monoisotopic molecular weight of the most abundant isotope. So I will personally prefer to use the average resolution. Okay, so I'll choose average resolution. So I click here to compute the PI molecular weight, which it will tell me about the theoretical PI value and the theoretical molecular weight based on my primary structure. So it shows here that the alcohol dehydrogenase will have a PI 6.97, which is very close to the physiological pH. And the molecular weight is about 39.5 kilodalton. Okay, so this is something that you have to find out before you outline any strategic planning for your purification. Okay, so here, okay, we know the molecular weight is about 39.5 kilodalton. The PI is 6.97, so around that. Okay, you can try with your own protein, okay, your own target protein of interest, the recombinant protein that you express. Okay, so you have the primary structure, you can easily compute the PI and the molecular weight too. So after we know about the PI, the molecular weight, what is next? Okay, let's go back to our talk. Okay, so now can you see the PowerPoint that I'm going to share here? Okay, so yes. So after we know about the PI value and the molecular weight, what is next? Okay, so what am I going to show you here is just some principles behind PI and pH. Okay, PI will be the pH of which a proteins carry 0 net charge. Okay, so when a protein carries 0 net charge, at the pH value, okay, that equivalents to the PI. So this is the isoelectric point of the protein. So why do pH, okay, affects the charge of a protein? So say when you have your PI value that is more than the pH of the environment, then the proteins will carry a positive charge. Okay, so remember this golden rule. When the PI more than the pH, proteins carry a positive charge. In contrast, when you have your PI value that is less than the pH, then the proteins will carry the negative charge. So how do we ensure that during the purification process, your protein, okay, is stable? This is very, very important because you do not want, after the whole series of purification, you get a denatured protein. Okay, so we consider the stability range. This stability range will be the pH range that is acceptable. Okay, there's a range of value that's acceptable away from the PI value to ensure that within this range of pH, your protein still retain its native structure. So it is important that we have to ensure this. But how do we ensure this? It is important, again, we have to use the buffer with appropriate pH to ensure the stability of the proteins during purification. All right. So what we would like to say here is, in protein purification, not only the temperature is important, but the pH is also utmost important. So to maintain the pH during purification, we rely on using appropriate buffer. Okay, so the best buffer will work when you have a pH plus minus the protein's PI value. So of course, if we consider a range, okay, a pH range to work with your protein, we often suggest a buffer choice, okay, having a pH value that's 0.5 to 1.5 units plus minus the PI value of your target protein of interest. Okay, so what I'm going to show you is an example of the protein purification, okay, if let's say you produce recombinant protein in bacteria. Okay, so this will be an outline of how you can prepare your sample. Okay, how you can prepare your sample when you have produced your recombinant protein in E. coli. So what you need to do, okay, the most common steps in sample preparation from cultured cells, okay, if we are looking at bacterial cells, are harvesting the cells. Of course, your cells will be cultured, okay, will grow in a culture medium. So what you need to do is you just need to recover the cells using centrifugation method. After that, whether or not you want to disrupt your cells, it depends on where is the expression. Okay, if your target protein is expressed extracellularly as secreted protein, this is rather straightforward because you can straight away take the culture medium, okay, and process from there. But the question is, how do you know whether your protein is expressed extracellularly or intracellularly? So you can run SDS-Bhd. So remember earlier on you have determined the molecular weight of your target protein of interest. So on SDS-Bhd, okay, what you can do is you recover the culture medium as well as the cells, okay, the bacterial cells, and then you run SDS-Bhd. And since you know the molecular weight of the target protein, when you run SDS-Bhd, you will be seeing, okay, if there's overexpression, there will be an intense band at the particular molecular weight range. So if your protein, okay, the band is very intense in extracellular, so you are very lucky. You get your protein expressed out from the cells into the culture medium. This is very straightforward to process. However, if the protein is expressed in intracellular, then you will have issue, okay? You need to do additional steps because you need to disrupt the cells, you need to lyse the cells to release the protein out from the cell. Okay, so intracellularly expression can be periplasm, it can be expressed in membrane, okay? Most of the time, the recombinant protein will express in cytoplasm. So what you need to do is you need to break the cell. So the cells must be lysed to release the intracellular materials, including your target proteins. So once we lyse the cells, we'll get the cellular debris. So this cellular debris has to be removed. It can be removed by centrifugation. It can be removed by filtration, okay? So for membrane proteins, if let's say your protein is expressed in membrane, then we probably need to do some solubilization, okay, to make your protein soluble. So I would like to emphasize here that it's very important that your protein has to be soluble before going into the purification step. All right. So if we look at the recombinant protein expression in the cytoplasm, the recombinant proteins can be soluble. This is pretty good, okay, because you don't need to take in another steps for solubilization. But if the target proteins are expressed as insoluble aggregates, which is known as inclusion bodies, then you need to take extra step whereby you need to solubilize this inclusion body, perform the refolding before going into purification. Okay, so we will talk about this in the next few slides. So if you notice here that there's a step called clarification. So the goals of clarifications are to prepare a clarified sample, which is a clear sample that must contain soluble protein for further purification. So it's important that your sample must be free from any particulates, or any contaminants that are not compatible with the purification. Okay, generally the samples for purification, okay, if we are looking at chromatography purification, it has to be, in general, be clear and free from particulates. All right. So the overall sample preparation requires a trade-off between the efficient extraction as well as the risk of protein denaturation. Therefore, sample preparation does need to be done quickly in lower temperature under the suitable condition, under the suitable buffer to reduce risk of target protein denaturation. So we have a question here. How do we know where our protein express? How to know whether they are soluble or not? Okay, I will talk about the solubility of the protein, okay, when we do the monitoring of the purity and functionality. But how do we know where our protein express? So as mentioned earlier, you can actually do an XDS page. Okay, so you collect the cells, the culture medium from your bacteria, for example. Then you run SDS page. So if there is overexpression of your recombinant protein, it should show as an intense band at the particular molecular weight. Okay, you know the molecular weight of your target protein of interest. So let's say it's a 35 kilodalton. If you observe an intense band at 35 kilodalton, let's say in the intracellular fraction, okay, then you know that your protein is expressed intracellularly. So during the centrifugation, you will get soluble supernatant. You may also get insoluble aggregates, okay? That could be your inclusion body. You run SDS page also on these two fractions. Then you will be able to find out where is the location of your expressed protein. So if it's in inclusion bodies, which means that your recombinant protein is not soluble, then we have to find a way to solubilize it. All right, we will talk about it more details in next few slides. So if you are dealing with eukaryotic sample, then you won't have issue with periplasm, okay, getting periplasmic sample, or you won't have issue with having the insoluble aggregates in your sample. So what are the purpose of sample preparation? Okay, the whole idea of sample preparation is we want to release the target protein of interest from the cells into the solution. If let's say your recombinant protein is expressed intracellularly. Secondly, we would like to remove any insoluble particles or impurities from the sample. This is important for the downstream purification because any insoluble particles can interfere with the purification. So the stability of your target protein of interest must be maintained. We have to ensure that the protein needs to be native. Okay, native means the globular shape of the protein still retained. And at the same time, the functions and the activity of the proteins must be maintained. So the conditions of your sample must fit into the chromatography purification. This is to ensure the success of purification. So again, I would like to remind you that all the protein extraction isolation should be performed quickly in cold condition in the presence of suitable buffer. So now we look into details how we can prepare the sample. What if the target protein is enclosed intracellularly? Okay, of course we have to break the cells. This is cell lysis. So to do cell lysis, we can have mechanical disruption. You can use sonicator, okay, to sonicate your cells together with appropriate buffer. Okay, we call it a cell lysis buffer. You can do homogenization, depends on whether you have this instrument in your lab. Okay, the common method that we use in our lab is a freeze-thaw method. So in freeze-thaw method, what we use is we freeze our bacterial cells, okay, using liquid nitrogen or in -80. Then we just thaw it at ambient temperature. So it will then break open the cells. You can also use enzymatic lysis where you add lysozyme, for example, into your sample preparation. If you do not mind, you add in additional proteins because lysozyme is an enzyme. Or if you don't mind, it add additional costs, okay, to your sample preparation. So the first step of protein isolation is always remember that you have to get your target protein out of the cells into the solution. So I would like to again emphasize here why buffer is required. So some of these proteins are meant for physiological functions in living organisms. So in living organisms, their functions and structures are well maintained according to the cellular pH. So the pH is regulated by the buffering system, as buffer can resist change in pH. So the choice of buffer is very important because the stability of our protein depends on the pH. When pH change, the charge of the protein will change. Okay, it is advisable that you use buffer that is compatible with your chromatography purification, because we would like to avoid any protein loss. Although we can do buffer exchange, okay, with protein concentrator, but still we will try to avoid that. All right. So some of the common buffers that we can use, like sodium phosphate 3-HCl, okay, you can use any buffer as long as the buffer pH is compatible and suitable to your target protein of interest. So another things that we can consider to add in is actually additive. You can add in additive. Additive is just like a seasoning you add into your sample preparation to make it flavorful. Okay, if you consider to add additive, you have to be careful and think about it. All right, be careful with the use of additive, because some additive are metal ion chelators such as EDTA. So if you are using EDTA, okay, it may have a bad impact on the protein, especially if you are working with enzyme. Some enzymes require metal ions as core factor. So you do not want the metal ions to be taken off from the enzyme, so the enzyme cannot function. Okay, so be careful when you want to add in additive. You can add in salt such as sodium chloride. Sodium chloride can be added to maintain the ionic strength, because ionic strength can affect the protein solubility. Okay, so be mindful also to use some reducing agents such as DTT or 2-mercaptol ethanol, because these two reducing agents can interfere with your purification step. Okay, and you do not want the reducing agent to overact on your proteins. You can also use detergents to solubilize the poorly soluble proteins. This is recommended, but bear in mind that you have to choose the mild detergent, okay, or you keep the detergent concentration as low as possible. You can use Triton or SDS, okay, but keep it at low concentration as possible to avoid protein denaturation, because this is not what you want. But there is one additive that you must add. Okay, I would recommend you to add a cocktail of protease inhibitor, because during the sample preparation, when you do cell lysis, it actually activates a lot of this intracellular proteolytic enzyme. So add in a protease inhibitors, okay, or a cocktail of protease inhibitor, okay, just like a proteinine, leucopectin, it can inhibit the protease, the enzyme. Okay, so some of these enzymes are serine protease, cysteine protease. So it's up to you how you want to strategize your sample preparation. But adding in protease inhibitor is highly recommended. Okay, so again, temperature, make sure it's under cold condition. So it may, some people actually conduct their purification in the cold room. I don't recommend that, because you won't be sitting in the cold room for the whole day. What you can do is prepare an ice bath. Okay, keep your sample, everything in the ice bath. Okay, so keep everything cold. It should be fine enough. So in some circumstances, the nucleic acid may have viscosity problem. So you can consider removing that DNA, RNA just by adding the DNAs or RNAs. Okay, so it all depends on how you want to prepare a sample. You need to strategize it. So after that, we can also filter, centrifuge, okay, to remove any insoluble particles. So if your proteins are associated with lipids, okay, you can use detergents or organic solvents to add in to your sample preparation to recover the proteins. After all, you need to ensure that your proteins must be soluble. So the choice of buffer, the temperature, pH is important to ensure protein stability. And the filtration and centrifugation is a must to prepare clarity samples, which is clear of impurities. So some people, they may do fractional precipitation. What I mean by fractional precipitation is we can add in salts such as ammonium sulfate. Okay, so the ammonium sulfate will increase the increased concentration of ammonium sulfate will reduce the solubility of proteins. So it starts to precipitate proteins. So when precipitate proteins, those unwanted materials will be get rid of. Okay, so it is also, you can also think of using fractional precipitation. It's fine. Okay, so if it's the salt, why is the salt important? So when you have ammonium sulfate together with your sample, what you need to do is you need to remove the salt. Okay, you can do dialysis, but I will not recommend dialysis because it will dilute your sample. What you can do is use a centrifugal protein concentrator. So you can actually find this information. There are a lot of different brands on centrifugal protein concentrator. At the same time, it do buffer exchange. It can remove salt and it helps to concentrate the protein. So the golden rules here is actually choose a gentle method to avoid protein denaturations. It happens when too vigorous cell destruction. Perform quick extraction in cold condition. Choice of buffer. So if your buffer, okay, you have to ensure the pH, the ionic strength fits into a target protein of interest, and wisely use the additive. Okay, the additive is like a seasoning to your sample prep. All the pH, temperature, and other conditions must be controlled to keep the protein stable. What I mean by protein stable is protein still retain its biological activity and the native structure, okay, the globular shape of the proteins remain intact. This is important. So as earlier we mentioned about inclusion bodies. So this is a species that occasionally present during bacterial expression. Okay, inclusion bodies refer to the accumulation of intracellular insoluble aggregates. Okay, inclusion bodies sounds like a treasure trove in recombinant protein expression. Of course, there are also challenges to isolate the target proteins. It becomes a big obstacle in getting active and soluble recombinant proteins. If they are inevitable, just accept them. Okay, why? Because the inclusion bodies often contain almost exclusively the overexpressed protein. And in inclusion body, the protein is protected from the protease degradation. And since the inclusion body is insoluble, okay, it actually helps in the isolation. So the general strategy of recovering bioactive proteins, okay, from the inclusion bodies would be, first, we must isolate the inclusion bodies. Since inclusion bodies are normally insoluble, we can just centrifuge it, then we get the inclusion bodies together with other contaminants. Then you need to wash it. Okay, washing is to get rid of the cell debris and contaminants. So the next step that is important in preparing inclusion body is to solubilize your inclusion body. So the solubilization of inclusion body, we can use mild detergent, okay, or any caltropic agent such as urea or guanidine hydrochloride. This will help to break open your inclusion body, okay? But at the same time also, your inclusion body will have those misfolded protein, okay, or insoluble protein. So we need to refold the protein. So protein refolding, okay, which means you introduce a mechanism to your inclusion bodies so that they can slowly fold back to their native state. Okay, so how to slowly fold back to their native state? Of course, you can perform some dialysis, like removing the solubilization solution. Okay, the solubilization buffer will be slowly removed. Okay, or you just dilute. Okay, so that to make your solubilization buffer to be less dilute, then the protein will slowly refold it. So the solubilization and refolding process has to be optimized. So another thing that you can think of is, if let's say your target protein contains disulfide bonds, you can add in redox agent to initiate the redox reaction, okay, during the refolding. So one good example of redox agent that you can add in is like glutathione. You can use oxidized and reduced glutathione in 1:3 ratio. Okay, so this will ensure that you will get back your disulfide bond. Okay, if let's say your target protein has disulfide bond. Therefore, it's very important you must understand the properties of your target protein. So usually the refolded proteins will be soluble. Then now you can go into the purification. Okay, so we will next move into the purifications of your soluble protein. So what I would recommend you to use is a chromatography technique. So chromatography technique, okay, in protein separation is based on the principles whereby there will be separation of mixture of proteins. So of course, chromatography is not restricted only to proteins. If you want to use chromatography to separate any compounds, the same principles apply. Okay, so we can apply the same principles as we want to separate different compounds in protein purification strategy. In fact, chromatography does more than just purification. We can use chromatography generally for two main purposes. First, for analytical purpose. Secondly, we can use it for preparative purpose. So in analytical, if let's say you want to determine the composition of your sample, okay, let's say the relative abundance or the absolute abundance by area under the curve or by absorbance of your protein, you can use analytical method. But in our context today, if let's say you want to purify sufficient quantities of target protein of interest, then we will use preparative chromatography. So comparing analytical and preparative, the starting amount of sample will be different. The column volume used will be different. What is column volume? So column volume refers to the resin that is packed within a column. So I will show you example. A typical protein chromatography has a column. It can be a plastic column, it can be a glass column. But inside this column, it will contain the resin that allow the separation to happen. So the separation is based on differential distribution between the stationary phase as well as the mobile phase. Okay, so when we say differential distribution, meaning that the stationary phase will usually consist of your resin that has certain properties that allow your target protein to interact with it. So any proteins that cannot interact with the stationary phase will be removed with mobile phase. Since it involves the use of mobile phase, we call it as liquid chromatography. So in protein purification, we have to use the mobile phase that is compatible with your protein with the stationary phase. So most of the time, the mobile phase used will be the buffer. So the purification using chromatography technique can be automated. Okay, such example shown here is actually an ACTA purification system from GE. Okay, very commonly used. Everything is automated. Okay, including loading of sample, the elutions. Okay, and also the collection is automated. But you may ask, if I don't have this, can I still do the chromatography? Yes, you can use a manual way. Okay, you just need to get the resin. You can even pack your own column. Okay, just using the syringe will do. Then you just manually load your sample and it manually elutes and collect your sample. Okay. So a general automated chromatography, okay, can be explained when you have a pump. This pump is known as a binary pump. Okay, the binary pump allow you to change the composition of buffer. So there will be two buffers used in protein chromatography, buffer A and buffer B. So the buffer composition can be changed so that it helps in the separation. So in an automated protein chromatography, we have a gradient mixer to help you to mix different compositions of the buffer. There is an injector for you to introduce and load the sample. and a column for separation to happen. So for protein chromatography, we will use a UV detector at 280 nanometer because protein absorb maximally at this wavelength due to the presence of tryptophan and thyroxine. So there will be a fraction collector for you to collect your sample at the end, and an automated chromatography recorder, okay, usually on the PC screen where you can observe the chromatogram. Okay, so what usually happens is you load your sample through the injector. Okay, different system will have different care and steps that you need to take care of. But generally, when you load your sample in, okay, the pump will start to pump the mobile phase to the column for separation. So we look at this example. When the red color sample goes into the column, it starts to separate into 3 different bands. Okay, so it will slowly go down to be detected by UV detector. And now you see here that the absorbance will start to go up. So the first fraction represents the purple protein is now appear in your chromatogram, and you can select and collect your purple protein. Same applies to blue color protein. Second fraction comes out. Last but not least, your green color protein. Okay, so this is an automated chromatography. Everything is automated. But even if you don't have an automated system, you can still do a manual chromatography. It's just that you need to do everything manual on your own. Okay, you need to collect your sample on your own. You need to measure the absorbance on your own. That's it. So the principles is still the same. So our focus today will be the purification of his-tag protein. Okay, why is it his-tag protein? Because his-tag protein is actually the most common tag that present in the recombinant protein. So now you have a pool of soluble protein sample that you can load into chromatography. Okay, in a typical affinity chromatography, the protein separation is based on the binding of protein to a specific ligand on a stationary phase. And usually this ligand is immobilized on the stationary phase. The ligand can recognize specific binding site on the protein. Such interaction can be biospecific. If let's say you want to purify immunoglobulin, IgG, then you can use protein A because it can recognize the IgG. Okay, the interaction can also be non-biospecific. If you want to, let's say, purify his-tag protein, then you can immobilize the nickel ion on the stationary phase. Okay, so we will say that affinity chromatography is high specificity due to biospecific interaction. So let's say when you have an impure sample that pass through this affinity chromatography, then the target protein will bind to the immobilized ligand, while other substances will be washed out from the column with the buffer. So now you can recover your target protein by changing the elution condition. Then it will displace the protein from the matrix. Okay, so the purification of his-tag protein is undertaken with similar principles, affinity chromatography. The separation of his-tag protein from other soluble proteins is thus based on the reversible interaction of your his-tag with the ligands on the stationary phase. Okay, again, when you have your his-tag protein, let's say expressed intracellularly, being in inclusion bodies or in soluble form, it doesn't matter because you still need to perform cell lysis. So once you lyse the cells, you get the his-tag protein. Okay, now you can apply a his-tag protein on a column known as IMAC. IMAC stands for immobilized metal affinity chromatography. Okay, I will explain this in a while. So your his-tag protein should be a string of 6 to 9 histidine residues. Okay, that is commonly used in the production of recombinant protein. So these histidine residues can be on your N-terminal or C-terminal. It depends, okay, whether you want to express either way. Either way will do, but try to avoid the functional domain. Try to avoid the functional site. Okay, you do not want your his-tag to present in your functional site. That may affect your functionality. Okay, so what happened is The purification of his stack protein happens when we can actually allow the binding of his stack on immobilized metal affinity chromatography. In IMAC, okay, what happens is there will be metal ions that are immobilized on the stationary phase. These metal ions generally are nickel. Okay, if you look at the resin, it's a bit light blue. It can be cobalt as well, okay, which is a bit of pinkish. So use a suitable binding buffer condition to bind the his stack on the IMAC, while the unbound protein will then be washed out. After that, you can just change the elution condition to elute the his stack protein. So you may ask, how does the purification of his stack protein work? So we use either nickel or cobalt. Okay, so how to choose between these two? Okay, what I show you here is the properties of the ligands. So you have to choose whether you want higher yield or higher purity. Since nickel ion has a higher binding capacity, therefore it will give you higher yield. But if higher purity is your concern, then you may want to use cobalt, because cobalt exhibits a more specific interaction with the histidine tag. Okay, so what happened during purification is the polyhistide protein will bind to the IMAC, okay, at the buffer having pH near to the physiological pH, so usually pH 7.2. You can add sodium chloride for ionic strength. So in the binding buffer, it is recommended to use low concentration of imidazole to prevent non-specific binding. Okay, low concentration can be about 25 millimolar of imidazole. And once your his stack binds to IMAC, you want to recover the his stack, just change the elution buffer by adding high concentration of imidazole. Okay, why are we using imidazole for elution? Because if you look at the structure of your amino acid histidine, histidine has an imidazole side chain. Okay, histidine side chain has an imidazole ring. So the imidazole can compete with the histidine to displace the his stack protein from the IMAC. So bear in mind that I would like you to pay attention, use high purity of imidazole, because imidazole will also absorb absorbance at 280. So there are some imidazole that has very low absorbance at 280. So use those imidazole because you do not want that to interfere with the absorbance reading for your collection. And one thing to take note is different IMAC product will have different product specification. So before you start, read the product specification carefully in the choice of buffer use and whether or not you want to use additive. What kind of additive is compatible with the IMAC? Okay, so the overall flow of his stack purification would be, of course, you must equilibrate your column with the binding buffer. Okay, you apply a sample, you apply binding buffer. So before you apply a sample, you need to make sure your column fill with binding buffer so that it's compatible with the purification. And now those unbound protein will be eluted out. So this broad tick here actually represents the unbound protein. Subsequently, you can change your buffer condition, okay, change to elution buffer, then it will elute out the his stack protein. And then you can re-equilibrate your column for the next purification. So what I show you here is actually a stack gradient elution. So you increase, okay, the buffer composition of elution buffer in a stack mode. So each condition can be probably 1 to 2 column volume. So you can see the x-axis here is actually the column volume. The column volume represents the volume of your resin. Okay, it can be 1 mil, it can be 5 mil. I will talk about the difference between these two. Okay, so of course you can also do a linear gradient elution, but bear in mind that to do linear gradient elution, okay, you must have 2 pumps. You can do a manual collection, but it is advisable that you have 2 pumps to adjust and alter your buffer composition to achieve this linear elution. So if let's say you have obtained the his stack protein of interest, okay, what you need to do, you need to define the level of purity and yield. One thing to take note as this, after each stage of purification process, the protein concentration will decrease. The more purification steps undertake, the more protein loss. So we must try to minimize the purification steps if possible to avoid protein loss. So if after affinity chromatography, you get your pure protein, then you are totally lucky and fine. You don't have to proceed with a second step of purification. Bear in mind that a good purification procedure should consider both purity and the yield of the proteins. And lastly, you have to check the purity of your target protein as well as perform bioassay to determine activity. So it depends on whether your target protein is an enzyme, then you can do enzyme activity. If it's a metal ion binding protein, then you can perform some binding assay to confirm the activity. So in order to define the level of purity and yield, the first question is, how do you want your protein to be? Secondly, how much of protein do you want? It depends on your downstream application. For instance, if you want to use your recombinant protein to produce the immunoglobulin, then the purity of your target protein has to be around 97%. So this is to try to get rid of impurities that could elicit undesired immune response in your host. If you would like to have your recombinant protein for biophysical characterization, such as NMR, crystallization, or circular dichroism, then the protein has to be 99% pure. If you just want your recombinant protein for downstream bioassay, then at least 95% purity is sufficient. But the yield will be of your concern if you need large amount of the recombinant protein for bioassay development. So by looking at the percentage, of course, we would like to have protein that is as pure as possible, at the same time as higher yield as possible. But how do we strategize this? So we can optimize around the binding and elution buffer condition. So in binding buffer, we can add low concentration of imidazole to avoid non-specific binding. While in the elution condition, we have to optimize the concentration of imidazole as well as the buffer pH, because the buffer pH will affect the interaction of the histag protein with the IMAC. Okay, so however, do note that if you add too much of imidazole to your buffer, it will reduce the yield. So you see, you want a higher yield, but the purity is low. At the same time, you want a higher purity, but the yield is low. So we have to optimize it. It depends on what you want at the end. So ideally, it can be here, okay? The purity is okay, the yield is okay. So it's totally up to you. So you may ask, if I can increase my sample volume to increase the yield, it depends on the resin binding capacity. So you need to check the product specification. It will mention that the binding capacity of the histag to the resin. Try to keep the sample volume according to your column volume. Usually we use 1/10 of our column volume. Let's say your column volume is 5 ml or 10 ml. Okay, if it's 10 ml, then you can add 1 ml of your sample. So if you have more sample volume, consider upscale your column that can accommodate your sample volume. If you load more sample than the binding capacity, the ion leakage can occur. So again, choice of metal ions, you want to use nickel or cobalt, depends on whether you want purity, you want yield or not. Okay, so of course, you can also play around with the elution pattern, whether you want to use a step elution or a linear elution. So whichever way will do. If you do not have an automated system, okay, you start with step elution. So let's say this is your histag protein and it's eluted at this, starting at this condition. Okay, so you have find out that this condition will be the optimal condition for the elution. So now you optimize it, then you can apply in your subsequent purification. Okay, so no worry, even if you don't have an automated system, you can always optimize it. It's just that you may need a longer time to do it, but it's feasible. So another thing that I would like to draw your attention is, if you want to improve the purity, okay, if let's say what we see here, very unfortunate, you get 3 bands, okay, you run SDS page, at the end you thought you get a pure protein, but unfortunately when you run SDS page, there are 3 bands here. This is not what you want. You thought you get a pure protein. So don't give up. You can still save this. You just need to add in another step of purification. Okay, so another way of improving purity is performing multi-step purification. Just add in a second purification step. In this case, I will recommend to use size exclusion chromatography. Okay, because you know the molecular weight of your target protein. Let's say in this case is 37. Okay, so you know this is a desired protein. These two are not. You want to get rid of this. So use size exclusion chromatography. Okay, select the fractionation range that remove these 2 proteins. Okay, let's say these 2 proteins having molecular weight less than 25 kilodalton. So choose the fractionation range, remove these 2 proteins, and you are able to recover back your desired protein. Okay, if multi-step purification is unavoidable, try to add just another 1 step, because the more purification steps that you add in, the more protein loss. Okay, remember this. So the next would be, do I need to remove his stack after purification? I think you have heard about this in the earlier talk today on the cloning, okay, whether you want to remove the his stack or not. So consider these 2 factors. Whether your his stack will interfere with the function of the protein, which means whether your his stack presence in the functional site of the protein, and whether the his stack will interfere with the protein native state. If yes for these 2, then you have to remove it. So his stack can be removed by sequence-specific protease such as HRC3D protease, PEV protease, thrombin factor XA. But bear in mind that such recognition site needs to be added to the vector before the expression. Okay, so if you do not have this sequence-specific site on the vector during expression, then after that you want to remove it is not possible. Okay. So we are a bit out of time, but we are almost reached to the end of our session today. What I want to show you now is very important after what you need to do after you get your pure protein. So starting from cloning, expression, okay, this requires some optimization. And then later on we have sample prep, purification, okay. Now, once we get our pure protein, we need to monitor its purity and functionality. You can undertake the following steps to monitor the purity and functionality of protein. First, SDS page. Use SDS page to monitor the complexity of your protein during sample preparation, during purification. You may want to know where is your protein. You may want to know whether your purification is successful or not. Last but not least, once you get your pure protein, you want to double confirm your protein is pure. So you can use native page. Native page is used to determine the molecular weight of soluble proteins after the purification, and you want to confirm the purity and functionality again, use native page. Soluble proteins will separate well in native page. Protein quantification assay, okay? During sample prep, during purification, determine the yield, okay? Determine the protein concentration. Protein concentration equals to the yield. And at the end, once you get the pure protein, you need to find out whether how much of proteins you get from the pool of sample. So since you are using his stack, so it's good to know that there are a lot of commercial antibodies available targeting his stack. So if you are expressing his stack in your recombinant protein, if let's say you want to do some immunoassay, you won't have issue in getting the anti-his stack protein. Okay, if let's say you want to do immunoassay targeting a recombinant protein and it has an his stack, so you don't have to purposely produce antibodies against your target protein. You just need to buy the anti-his antibody that it can bind to the his stack. So you can use Western blot, okay, use anti-his antibody. You can detect the presence of your his stack protein as well, okay, during sample prep, during purification, and whether after the purification is done. So of course, the actual confirmation of your target should be performed by mass spec, or N-terminal sequencing to confirm the protein identity. Last but not least, it's important that you carry out some bioassay to confirm activity, because after all, you want your protein to retain its activity at the same time it's pure. So if you're dealing with some downstream application, okay, bear in mind that you have to ensure your pure protein, the buffer composition of a pure protein has to be compatible with downstream application. If it's not compatible, no worry, you can do buffer exchange. I will not recommend you to use dialysis, okay, because it will dilute a sample. Use a protein concentrator. It helps to do buffer exchange at the same time in concentrate your protein. So there are a lot of different brands of protein concentrator out there. You can have a look on those protein concentrator. Select the most appropriate protein concentrator for your protein. So different types of protein quantification assay you can use. So UV is not meant for actual quantification. UV is just for you to detect whether there are proteins present or not. So basically, you can straight away measure your protein at 280. So if there is an absorbance, meaning there are proteins inside. But to be precisely determine your protein concentration, please use some calorimetric assay such as BCA. BCA is actually a modified of burette. You can use Bradford, you can use Lowry, okay? But do bear in mind that detection limits, okay? And at the same time, you have to ensure that your calorimetric reagent is compatible with the buffer of the protein. So after you get your purified protein, how do you store it? You can store it in -20. It's sufficient to store your active protein. But do bear in mind, keep your protein sample in small aliquot so that avoid freeze thaw, okay? So this will be the second last of what I want to share with you, okay? You can make this table to monitor your purification performance. What is meant by purification performance is you want to ensure you get a yield, a good yield, and a pure protein. So you can make a table like this. So every steps that you undertake in the purification strategy, just list it here, you can perform some protein quantification assay, determine the amount of the protein. Of course, you need to perform some bioassay, determine the activity of the protein, okay? After that, determine the specific activity. So specific activity will be the total activity over the amount of the protein. Then later on, you can quantitate the yield. The yield will be the percentage, okay? The total activity in every step divided by the total activity during the initial sample prep. It will tell you the yield of your protein, and the purification level will tell you how pure is your protein. So this table has been widely used in purification of enzyme, but you can use it for any purifications that you want to, okay, to monitor your purification performance. So I just want to share with you an example on how you can outline your recombinant protein from cloning up to the purification. So this is an example of this group of researcher, Kim, okay, in 2011. They would like to produce a recombinant natural killer cell enhancing factor. This NKEF exhibiting anti-proliferative activity as well as antioxidant activity, okay? So start with cloning, expression, okay? Later on, they resuspend the BL21 bacterial in guanidine hydrochloride, the caltropic agent. So which means that the protein is actually retained probably in their inclusion bodies, okay? So add some imidazole in so that it's compatible with the subsequent nickel purification, okay? So do some sonication, centrifuge, okay? These are all the normal purification procedure. Then perform SDS batch, okay? So lucky, it's very lucky that get the pure protein after just one purification. Then perform Bradford assay to determine the protein concentration. And as mentioned earlier, this protein has anti-proliferative and antioxidant activity. So perform these two bioassay, okay, to confirm that the protein that is expressed is active. Okay, so this protein has a molecular weight about 25 kilodalton. Okay, So we have reached to the last phase of our session. Sorry, we have gone beyond the time, but it's good that I have to show you this. Use this checklist, okay, to assist your protein purification workflow. First, you must understand the properties of the target protein and select the most appropriate sample preparation methods. Optimize it, okay? If you're dealing with recombinant protein, if your protein is soluble, insoluble, what will be the preparation that you need to think about? Consider the chromatography resins, okay, for purification. It's important that you check the product specification, check the compatibility of your sample with the chromatography resin, and then you optimize the chromatography workflow, optimize your binding and elution buffer. Then your downstream application, you want the purity more than the yield or the yield more than the purity, and monitor your purification performance. Last but not least, you get your final product, confirm the purity, perform bioassay to confirm its function, and conduct sequencing or mass spectrometry to confirm its identity. So if you are using bacterial in expressing your protein, and if your protein requires some post-translational modification, okay, but unfortunately, this cannot be achieved in bacterials. What happens is you may want to opt for in vitro post-translational modification. Okay, we have come to the end of our session today. Thanks for staying back with us. Before you go, I would appreciate if you can also provide a feedback to this session. I need to add protease inhibitor. Okay, this is a very good question. So the protease inhibitor, okay, is meant to inhibit the intracellular proteolytic enzyme, because this protease may also cut your protein. Okay, so if your target protein itself is protease, I will still recommend you to add protease inhibitor. But later on, after your sample preparation, you want to recover your active protease, then you do a buffer exchange to a buffer without protease inhibitor. Then it will get rid of the protease inhibitor that will inhibit your protease. All right. Another question would be, are nickel and cobalt universal? Can they be used as binding ligand for other tag? What about elution buffer with imidazole? If the protein of interest is tagged with different tag, do we need to use imidazole rich buffer? Okay, nickel and cobalt are commonly used for his-tag protein purification. So if let's say you tag your protein with GST, for example, okay, I think I missed out this in my talk earlier on. If you decided to tag your protein with GST, GST is an enzyme. So you have to be careful when you work with enzyme. You have to take care of your protein, you have to take care of the GST. If the GST gone, then your purification will be a problem. Okay, so if you want to use GST tag, then of course the imidazole won't be the elution buffer. Then it will be a glutathione. So when you choose a different tag in your purification, try to read up, okay? Try to select the correct chromatography resin, and you read up the chromatography resin product specification. So inside the product specification, the manufacturer will tell you what will be the optimum condition that you can use for your binding and elution condition. Okay? If I answer your questions correctly, if I understand your question. Okay, is there any more questions coming in? Okay, if no questions, yes, we will try to work out with our co-host to see how we can share this talk video with you so that you can replay. Okay, at the same time, we appreciate if you can fill out the link, okay, not only for my session, but also for Dr. Patrick's session, so that we can improve our session. And in view of the current COVID situation, in fact, we would plan to actually have a hands-on workshop for about 2 to 3 days. Then it will be clear that every one of you will attend a talk like this and then some hands-on following that. But unfortunately, we have to postpone it. We will try to have it soon, okay, by monitoring the conditions. So hopefully you can join us in future if there's any hands-on workshop. Okay, so I hope to see you guys in future. Thank you. Thank you so much. yeah, appreciate your time to fill out the feedback to us so that we can improve. Thank you so much for your time. Thank you. See you guys again. Hope to see you.
Thanks everyone for joining. We are really happy to have all of you here. So hopefully, if we were to have the hands-on workshop, hopefully we will see you guys again. So thank you everyone. Thanks Dr. Michelle, and also thanks to the co-organizers as well.